Thanks for joining. Just letting a few more folks join and then we'll get started. All right. Well, welcome everyone to our webinar, 11th webinar with the International Oak Society and Global Conservation Consortium for Oak. I'm Amy Byrne. I'm at the Morton Arboretum, who leads the Global Conservation Consortium for Oak. And I'm also part of the International Oak Society board. The slide I'm showing here is just acknowledging the institutional members that we have a part of the International Oak Society. So we just like to recognize their support. So thank you. And thank you all so much for joining us today, taking the time. I know some people are joining really, really late in the evening, so we are very grateful for you attending. And so today we have two wonderful presentations in store for us. First, we'll hear from Dr. Yong-Shik Kim, who is Professor Emeritus with the Department of Landscape Architecture at Yeungnam University in Korea. And he'll share a presentation on the oaks of Korea. So very exciting. And then we'll have some time for Q&A following Dr. Yong-Shik's presentation. And then following that presentation and questions, we'll from Jesse Parker, a research specialist at the School of Natural Resources with the University of Tennessee in the United States, and he'll present on his master's research investigating hybridization and population genetics in trailing range edge populations of swamp white oak. And then, of course, we'll have some time for Q&A after his presentation as well. But now I'm going to pass it off to Roderick Cameron with the International Oak Society, who will share some announcements.
Hello, everyone. Welcome. Great to see you all here. For those who don't know me, my name is Roderick Cameron. I am the chair of the Tours and Events Committee for the International Oak Society and also the secretary. After our presentations, if you have any general questions about the International Oak Society, we would gladly field them and try to answer them. So keep those for the end. Just a few announcements before we start. The newsletter, Oak News and Notes, is about to go out, probably straight after this webinar. So watch your inboxes for that. And there's some announcements about events. You may recall we have Oak Open Days scheduled for May in the UK, 16th and 17th of May, and registration for that is just open. So you'll see in the newsletter a link to the announcement on our events page, and there a link to the registration page. There's information there. If you have any questions, just shoot us an email. The two other events are shaping up. The ones we're having for fall, Northern Hemisphere fall, that is. The tour of Southeast USA, we're finalizing plans for that, and we hope to open registration in March. So we'll announce that when that happens. And the tour of Turkey now has its own website. So the link for that will be in the newsletter. So I do recommend you have a look at that, because tour leader Elif Denise Ülker has done an excellent job producing a very informative website, not just about the tour, but also other things you can do in Turkey while you're there. So I think those are the announcements from me, Amy. So back to you, and I think we can get started with the presentation.
Great. Thanks so much, Roderick. Yep. All right. So yeah, first up we have Dr. Yong-Shik presenting on the oaks of Korea. So let me get that pulled up. Yong-Shik, does that look good to you? Everyone see my screen? Oh, you're muted, Yong-Shik. We can't hear you. You have to unmute. We can't hear you yet. So turn on your microphone. There we go.
No problem for me. Can you
Okay.
hear me?
We can hear you.
Yes, we can hear you now.
Okay. Mm, I'm sorry. Yes. Hello, good afternoon, and good morning. I'm very much uh, pleased to talk about on genus Quercus in Korea. Next, please. Next.
Yeah.
Yes. Due to temperature differences, the Korean peninsula is divided into five categories. The major mountain ranges are the Taebaek and the Sobaek Mountains, and this geomorphology defines the major vegetation types and flora. The two photos on the right side oak trees that were designated as natural monument in the early 1960s. In Korea, five oaks, including one groves, are designated as natural monument. Next, please. The Korean Peninsula hosts six deciduous and five evergreen oak species in South Korea. Furthermore, three additional species, Quercus oticifolia, Quercus crispula, and Quercus donarium, are known to occur in North Korean territory, and this could be included in the Korean oak flora. The frequent occurrence of natural hybridization among these species underscores the high potential for oak collection and research in Korea. Next, please. Here you can see how Korean oak species are distributed by altitude. Quercus mongolica grows at the higher elevations, while Quercus mucinifolia is found at the almost sea level. Next. You can see the map of North Korea and South Korea on the left side. Due to the limitation of information, I could not designate counties. So I designated only provinces in North Korea, while I designated the county levels in South Korea. The colored portion is where the herbarium specimens are collected so far. The white areas is not has no information on herbarium specimens. For example, Quercus dentata is widely distributed from North and South Korea, except highlands in the north of North Korea. The next one, next please. This is Quercus mongolica. It's one of the dominant oak species in Korea, distributed from lowlands to almost 1800s. It is moderately shade tolerant, allowing regeneration on the forest canopies, but shows enhanced growth with increased light availability. The species prefers well-drained soils and is highly adapted to cold climate, making it especially common in central and northern Korea. Next, please. This is information on Quercus elena. This species is dominant in oak species in Korea, found from lowlands to mountainous areas. It tolerates moderate shade, but grows better under bright conditions because it is adapted to cold climate. It is most common in central and northern Korea. You can see you can see the food in the bottom of the middle part. This is food from acorn. It is a traditional Korean food. Next, please. This is Quercus serrata. It's also common deciduous oak species in Korea, mainly found in the lower elevation and the low mountains. It prefers well-drained slopes and valleys and tolerate poor soils. As a light demanding species, it often dominates secondary forest. Next, please. Quercus acuta is limited in the southeast peninsula, a very narrow distribution, including Jeju Island, one of the biggest islands in Korea. It grows in lowland to hilly areas and well-drained soils and is more shade tolerant than deciduous oak species. Next, please. Quercus gilba is distributed in southeast Korean peninsula, especially in coastal areas and on islands. It grows in lowland to lower montane forest and is relatively shade tolerant. Next, please. Quercus glauca is mainly found in southern Korea also, especially along coastal areas and on islands. It grows in warm temperate climate on well-drained soils. This species is relatively shade tolerant and can grow well on the forest canopies. Next, please. Quercus macinifolia is mainly found in southern Korea also, including coastal areas and islands. It grows well in warm, humid climate and prefers well-drained soils. This species is quite shade tolerant and can grow well on the forest canopies. Next, please. Also, this species is widely used in landscaping trees in Korea. It has been misidentified as Quercus glauca or Quercus salicina. Field surveys show that natural populations are very limited, with only 169 mature individuals in three subpopulations on Jindo Island. Next, please. Quercus salicina, found in coastal areas and islands too. It grows in lowland to hilly forest on well-drained soils and can tolerate shaded conditions very well. This species is commonly associated with evergreen broad-leaved forest and plays an important role in maintaining a stable forest structure and year-round canopy cover. Next, please. This map highlights the records for this species in Korea, because you can see many areas with white colors, indicating the need for additional field surveys. The photo shows the species' natural growth form. While oak harvesting is limited today, oaks have historically played an important role in Korean forest. Next, please. Quercus variabilis is widely found in forest and rural landscape. It is valued for its strong wood, traditionally used for fuel and tools, and for its acorns, which are an important food source for human and wildlife, and were historically used by people. Next, please. Species level information from natural habitat in North Korea is severely limited, highlighting the need for collaborative research conducted beyond North Korea. Next, please. The figure illustrated natural hybridization among 4 oak species: Quercus serrata, Quercus elina, Quercus mongolica, and Quercus dentata. Each species can easily hybridize with the others, forming multiple two-way hybrids in the wild. These hybrids overlap in the center, indicating frequent gene flow among species. The diagram shows that oak species in Korea are connected through extensive natural hybridization. Next, please. This is cultivars in Korea. One is that Quercus dentata, Carl Perez Miller. Unfortunately, this cultivar is not well known in Korea so far, only known in European countries. The other cultivars, Quercus variabilis Johnson, was recently named at Charlie IV Arboretum. So we have only 2 cultivars from oak species in Korea. Next, please. Korea has made significant progress in oak research last year, with current effort increasingly oriented toward international collaboration. Enhanced cooperation at global, regional, and national levels is expected, with the Morton Arboretum, the Global Conservation Consortium for Oak, and the International Oak Society serving as co-partners in these initiatives. Next, please. This represents a preliminary database of native oak species I made in Korean Peninsula, which will be expanded and updated to include detailed species information, research bibliographies, and collection records. I hope you may visit this site and comment to make further development. Next, please. Summarizing my presentation, as a biogeographic crossroads for oaks, Korea is central, I think, to understanding East Asian oak diversity. Exceptional taxonomic complexity driven by hybridization and a taxonomically challenging system is ideal for testing integrative approaches. And also conservation significance beyond species count. We need for an integrative forward-looking framework, for example, systematic germplasm collection, herbarium-based revision, and also access to conservation. Next. Thank you so much indeed for your kind attention.
Wonderful. Thank you so much, Yong-Shik.
My pleasure.
Wonderful presentation. Thank you so much. We have time for questions. Does anyone have a question for Yong-Shik? Feel free to unmute and ask.
I can start off with a question. Well, I first want to start with a comment. I don't know if anyone noticed the date on Yong-Shik's cover, but it said 26th of February. That's not a mistake. It's
Yeah,
midnight
yeah,
for him. So
yeah,
he's doing it on the 26th of February. So I really appreciate that, Yong-Shik, the effort you've made to present live. Thank you very much. I have lots of questions, but I was very interested to see the amount of hybrids you suggested existed. They seem to
yeah.
be mostly in section Quercus. Is that
Mhm.
the case then? Or are the hybrids only recorded in that section? And my other question is, have they been named? Do they have their own epithet? And do you have also, you suggested there may be three-way hybrids as well. Have those been cataloged and named?
Yes, we have information, but we have very difficult situation because one part uh, could not admit, cannot can't admit, and the others still have difficult to say. There are many hybrids in the wild, and some are named, and some are not named so far, because the research on oak in the wild is very limited. So we are not widely surveyed. It was until 1980s, one professor who passed away has extensively investigated in the field, and many herbarium specimens are documented at the University Herbaria. Since after, just one or two scientists work that, not so extensively. So the naming of hybrids are still dormant until 1980s. But
Hmm.
I think we need work much more.
Sure, sure. I should point out if anyone wishes to put questions in the chat, we can take them there, Amy, as well, right?
Yep, yep. I see Brian has a question, and then there's one in the chat. Oh,
Okay. Thank you,
Yeah.
Yong-Shik.
Thank you so much for that wonderful presentation. I'm curious of those species you mentioned. Uh, can you speak a little bit about urban tolerance?
Abi,
urban. Like how do those species do in urban environments, like in cities?
urban environment.
Yeah.
Yes, that's a big problem because, as you may know, urban areas in Korea are rapidly expanded. That means fragmenting or destructing the wild habitat. For example, in Seoul areas, you can see many wild Quercus, for example, Quercus aquatissima, Quercus serrata, but the population are severely distracted to build the apartment house complex. So I think we need to collect germ plasm in every places to conserve the genetic diversity.
Great. Thank you for the question. Thank you, Yong-Shik. So there was a question in the chat about I think it was what was the relatively thin oak leaf with variegation on the
Yes.
slide showing the cultivars, which I think they're asking which species that was,
Uh,
the name of
let
it.
me see. Let me see.
I think it was Quercus variabilis.
Yes. Mm, yes. Quercus variabilis and Quercus dentata.
Great. Thank you. Thank you for the question. Kaula, do you have a question?
Hello. Can you hear me?
Yes.
Okay, excellent. So thank you, Mr. Young, for this impressive presentation. It was so wonderful to attend it. Actually, I am curious about the about due to the diversity that you present of the Quercus in Korea, and as previously I had some information about the socio-economic state situation of Quercus in Korea. I didn't have a clear idea about it, but I need to, I get some information that there's some impressive markets there about of econs, and I want to know uh, the socioeconomic state of econ there and the form of consumption. It's a little bit not in the topic that you talk about, but I am it's I found it I found it I would like to know about it. Thank you.
Amy, please help me because I could not fully understand.
Yeah, no problem.
Mm.
So, Kello's wondering or is curious to know more about the socioeconomic status or importance of oak acorns. So you
Hmm.
mentioned acorns being used to make food or for other
Yes,
products. So she's just
yes,
curious to know more about that.
yes. And acorn is a very good material for jelly, and the jelly is a traditional food from acorns. But
Mm-hmm.
the papers can awaken the importance as a food for wild animals.
Mm-hmm.
So
Mm,
the papers concerned about the collection of acorns in the wild.
Mm-hmm.
So I think it's very good uh, changes to conserve the nature for acorns in
Mm-hmm.
the wild. It's okay
Okay.
for.
Yes, yes, it's okay. So actually, I was wondering about the market of econ there. Is there any market of econ-based products in Korea?
Yes, still it's a very good business in the
Mm-hmm.
market.
Okay, thank you.
My pleasure.
I have a follow-up on that, Yong-Shik. You
Yeah.
mentioned the acorn jelly only under Quercus aliena. Is
No,
it
no, no.
it's other
Quercus.
species as well?
Yes, yes. Oh,
Okay. All section Quercus acorns.
yes, yes. Four
Yeah.
species.
Okay.
Great. Thank you for all these questions. I think we have time for one or two more. Any other questions?
Am I allowed another one?
Sure.
I was interested. You mentioned something about the difficulty with about finding information about North Korea. Could you tell us a bit more? I mean, you show maps where you show the occurrence. Is that based on very old field trips or herbarium specimens, or what sort of information are you able to get at the moment?
Yes, the map I showed you just before was collected information from the book literatures from printed in North Korea,
Ah,
and but they did not record it by county basis, just the
mm,
province basis. And last year in October, when I visited botanic gardens or arboretum in Eastern Europe, especially Czech and Poland, I saw many herbarium specimens for oaks, and I realized that we need to collaborate because, for example, the herbarium specimens of Calyx mongolica looks quite different than I expected. So I asked them to work together, and they said yes. So I hope we can work together with Czech and Poland.
great.
Great. Thank you for the question. There was just a comment in the chat. Jessica Goller from Chicago Botanic Garden was mentioning they have Quercus dentata, the cultivar Carl Ferris Miller at the Chicago Botanic Garden, and it's growing well, and it's one of her favorites. So I just wanted to mention that. So, all right. If there aren't any other questions or comments, thank you so much again, Dr. Yong-Shik Kim, for your amazing presentation on the overview of Oaks of Korea. It was really, really insightful and wonderful to see. And just thank you so much for staying up so late to present. And for those who joined us also, some of your colleagues who joined us as well from Korea, we're very grateful. So thank you. Thank you so
Thank you
much.
so much.
All right, wonderful. Thank you everyone for those questions and discussions. So now we're going to move on to the second presentation, Jesse Parker. Whenever you're ready, you can share your screen.
All right, let me this works. Can you see that?
Yep, looks great.
All right. Okay, so thank you all for tuning in to this, and thank you, Dr. Kim, for that presentation. I'm really excited to be here sharing this work with you all. So the title of this talk is Introgression and Genetic Variation at the Trailing Edge of Quercus bicolor. And this work was part of my master's thesis at the University of Tennessee, which I just recently finished up. So this work actually started as an applied germplasm conservation project in collaboration with the UT Tree Improvement Program. And it was aimed at understanding and conserving Quercus bicolor occurring in the state of Tennessee and developing seed orchards for the production of plant material for reforestation. But what I will actually primarily be talking about today is a bit different from that, being focused on the population genetics and hybridization, which sort of was insights that came out of that project. So I will periodically reference how these concepts relate to the ex situ conservation throughout this talk. So some of these findings were recently published under this title. So if you want more details, you can check that article out. So to give a brief outline of this talk, I'm first just going to introduce the focal species, Quercus bicolor, give a little background on it. And then I'm going to discuss some of the importance of genetic diversity and introduce this concept of the central marginal hypothesis, which is a major part of the hypotheses that guided this research. And then I will get into my specific research questions and some of what I found. So the species I am primarily focusing on today is Quercus bicolor, the swamp white oak commonly. And it's a beautiful species of white oak native to the eastern United States and just barely extending into Canada, but primarily the eastern US. Here are a few pictures of swamp white oaks from various parts of its range. So swamp white oak is a member of the white oak group in the Quercus subgenus Quercus section. And in this particular topology from Larson et al. 2024, you can see Quercus bicolor at the top there, circled in red, followed by Quercus lyrata, the overcup oak, as its closest relative, and then next down is Quercus michellii, swamp chestnut oak, and then other close-ish relatives include bur oak, macrocarpa, lobata, stellata, muhlenbergii, montana, and alba. So all of these eastern North American white oaks. So some of the morphological distinctions of this species include having an especially long peduncle, which attaches the acorn cap to the branch. And the specific epithet bicolor comes from the whitish pubescent below leaf surface when compared to the dark green upper leaf surface. As suggested by the common name, it is a bottomland species generally occurring on hydromorphic soils characterized by periodic inundation. In field trials, it has shown some superior flood tolerance when compared to most other hardwood species. Here you can see the alleged natural distribution with these light green counties being where the species has been reported. It is generally considered uncommon throughout most of its range, though it is a characteristic component of several forest types, primarily in the Great Lakes and Northeast regions of the United States. Acorns of swamp white oak are notably low in tannins and highly valued by wildlife, especially ducks. And like other oaks, these trees support numerous arthropods, making them foundational within ecosystems. Swamp white oak is also a valuable tree for timber, generally being marketed as simply as white oak. And in recent years, swamp white oak has gained quite a bit of popularity as a landscape tree. In this picture on the right, you can see the 9/11 Memorial Plaza in New York City, where they've planted over 400 swamp white oaks. You can also see it commonly on streets throughout cities in the U.S. So now before I get into my specific research, I want to just mention the importance of genetic diversity. So genetic diversity is directly correlated with the adaptive potential of populations and species, and it is maintained in natural populations through these processes such as mutation, recombination, and migration. Some of these alleles and combinations of alleles will be deleterious, others may be advantageous, and many others will be neutral currently, but could become advantageous with changing conditions in the future. So conserving the full range of diversity within a species, including unique variants within populations, will increase the likelihood that these advantageous alleles will be available to allow a species to persist through environmental change. So genetic diversity is also correlated with population sizes. So small isolated populations are often less diverse than large interconnected populations. So genetic diversity is not constant within a species across the landscape, as it will tend to co-vary with population sizes and environmental factors. For example, genetic diversity can vary across the landscape as a function of position within the range or distribution of a species. So this gets into this concept of the central marginal hypothesis, which posits that species should be most abundant within core parts of their range, as this is where their adaptations most closely match the environment. So at range margins, populations often become smaller and more isolated, as illustrated in this conceptual depiction of a species range on the right, as well as in this depiction of the natural distribution of swamp white oak. So the reduction in population sizes at range margins is thought to be due to increasing levels of phenotype environment mismatch, resulting in reduced fitness and ultimately being realized as these smaller and smaller populations with increasing fragmentation. So if true, it is thought that this phenomenon should be measurable through DNA analysis as reductions in within population diversity at range edges and increases in among or between population diversity, also called differentiation at these range edges. However, numerous exceptions to these trends exist. For one, historic demographic events or migrations can complicate these underlying assumptions. For example, some species show high genetic diversity in areas that served as historic glacial refugia that are now in range marginal positions. Additionally, interspecific hybridization can increase at range margins where species that are normally allopatric come into contact, or demographic imbalances or environmental factors can favor hybrid formation and survival. Hybridization can result in elevated levels of genetic diversity in contrast to these expectations and allow populations to persist or even expand despite marginal environmental conditions. through the transfer of adaptive alleles. So the propensity of interspecific hybridization to increase at range margins has not often been rigorously tested, even in taxa known for high hybridization potential, such as oaks. Often the evolutionary importance of hybridization is downplayed or thought to be part of range limiting processes by assuming hybrid inferiority. So range margins or edges can be further classified into leading and trailing edges, referring to the movement of a species over time as climates change. So in this example on the right, this species is moving north through time, a trend generally observed in many North American and European species following the last glacial period, leaving behind these relict populations in the south, termed the trailing edge, while in the north, colonization of new territory is occurring, termed the leading edge. So for conservation, the idea of preferentially conserving rear or trailing edges has been touted as a possible avenue to improve resilience of species to climate change. Basically, the idea being that trailing edge populations are better adapted to warm conditions due to their position at the warmer edge of the species range. The transfer of trailing edge genes to northern parts of the range through assisted gene flow or incorporation into breeding programs are strategies that could be used to enhance adaptations. However, because many of these populations are also range marginal, they may have reduced genetic diversity, increased population structure, or increased levels of introgression, all of which can complicate conservation. For example, increased population structure might indicate local adaptation, which should be accounted for in ex situ conservation plans. Additionally, hybridization at range margins can result in local extinction or the loss of species-specific alleles through genetic swamping, and can also alter the ability of a species to respond to climate change through habitat tracking. Additionally, the presence of unknown cryptic hybrids in populations of interest is also a major problem, as practitioners might simply not be conserving what they say or think they are conserving taxonomically. Thus, understanding the actual distribution of genetic diversity and rates of hybridization by genotyping individuals across a species range is an important prerequisite to initiating conservation actions, particularly at range margins. So in light of all of these considerations, and given our interest in developing trailing edge germplasm conservation resources for swamp white oak, using population genomics, for this study, I set out to address these three kind of broad questions. First, is there more introgression at the trailing edge compared to the core, as might be expected in a genus such as Quercus? And then, is there more differentiation, and is there less within population genetic diversity at the trailing edge compared to the core, as might be predicted by the central marginal hypothesis? So to address these questions, I had designed a DNA study using RAD-seq sequencing methodology. I first extensively surveyed the trailing range edge using historic records and species distribution modeling to locate populations. I then collected leaf samples from all of the mature trees found at the trailing range edge in Tennessee, as well as from 9 populations across the core of the range, designated as such through species distribution modeling. So these populations, these pink triangles in the north, are what I mean when I refer to core populations later in this presentation. So after genotyping these individuals, the first area I explored was introgression, which refers to the movement of alleles from one species to another through hybridization. Hybridization and introgression are important to consider when implementing ex situ conservation actions, as inadvertent inclusion of hybrids into seed orchards could compromise management objectives. That said, introgression can also have evolutionary benefits by transferring adaptive alleles and potentially allowing new combinations of genomic elements to be tested. So we probably all know that in Quercus, hybridization is known to be common. For example, in this image on the right, we have a classic depiction of the white oak syngamion from Hardin 1975, where each circle is a species and each line is a documented natural hybridization event. So in this syngamion, species appear to freely interbreed, yet appear to remain distinct. So in this, you can see that Quercus bicolor with the arrow is known to hybridize with chestnut oak, southern live oak, post oak, white oak, overcup oak, and bur oak. That was kind of going in counterclockwise order. Other than scattered reports of such hybrids, mostly from the early 20th century, no further information on hybridization and introgression in swamp white oak exists. So in order to evaluate levels of introgression, I acquired additional DNA samples from another study, Hip et al. 2020, for these selected species, which consisted of sequence reads from 2 to 6 individuals for each of these species. And of those known to hybridize, only Quercus virginiana was not included, as these do not occur sympatrically in the study area. So here's what I found after some initial analysis. This is the principal components, component analysis biplot on the genomic data. And you can see the reference samples for these 5 species on the right, clustering fairly well together. And then here on the left, you can see this dense cluster of my samples, and then the swamp white oak, the Quercus bicolor, and Quercus lyrata reference samples that I included. So this suggests potential gene flow. And then looking a little deeper, you can see in this structure style plot where each vertical bar is an individual and the colors indicate ancestry components. In the center of the plot, these entirely light blue bars are the samples collected from the range core, which are clustering with the swamp white oak reference samples kind of on the right side where there's those light blue bars, essentially indicating pure swamp white oak ancestry. In contrast, the Tennessee populations on the left have this clear pattern of introgression from this green ancestry component, which corresponds to the Quercus lyrata reference samples over on the right. So essentially, the Tennessee individuals appear to be occurring along a spectrum between the swamp white oak and the overcup oak. And that was found in nearly all of the Tennessee populations. So this level of widespread hybridization between these species is a pretty exciting discovery, which warrants more investigation. So as seen here, I also found a clear relationship between the level of admixture and both the individual level heterozygosity on the left, as well as FST, a measure of between population differentiation on the right. So essentially, as admixture increases, going from right to left on the plot, genetic diversity and population differentiation also increase. Ultimately, this is a somewhat expected result, as hybridization is expected to increase heterozygosity, but it does highlight that what might otherwise be genetically impoverished range marginal populations are gaining genetic diversity and potentially adaptive potential through interspecific admixture. So to summarize these findings relative to this question of introgression, I found no evidence of introgression in the core populations, while in the Tennessee populations, I just detected widespread introgression, specifically from Quercus lyrata. And I found evidence for both early and advanced generation hybrids and strong relationships between these levels of admixture and measures of genetic diversity and population differentiation. Interestingly, I also found evidence of introgression in some areas of the trailing edge where overcup oak is not known to occur anywhere near there. So on that last point, I hypothesized that historic climatic conditions since the last glacial maximum could have possibly permitted a broader overlap between these species, thus explaining this signature of introgression. So to explore this, I built a species distribution model for both of these species and used backcasted climate projections for 22 and 6,000 years before present to predict how the hybrid zone could have shifted since the last glacial maximum. And I found that these species were likely restricted to much smaller ranges in the coastal plain during the last glacial maximum, but could have spread to much larger areas in their current distribution during the mid-Holocene, 6,000 years before present. So this could explain a persistent signature of introgression, even in swamp white oak populations that appear disjunct today. I also projected these models into the future, showing that this hybrid zone is likely to become more significant for both of these species with climate change. So now that I established that this introgression is occurring, the second area I wanted to address was population differentiation. And specifically, I asked, is there more differentiation at the trailing edge compared to the core, as predicted by the central marginal hypothesis? So first, I removed the highly admixed individuals using a threshold applied to the structure results. And then I assessed population differentiation and structure using admixture analysis, which detected a greater number of unique population clusters in Tennessee, as seen in this figure. And also pairwise and overall FST values were greater in Tennessee. And analysis of molecular variance, or MOVA, found that a greater percent of variation was explained by between population differences in Tennessee when compared to the core. So overall, this suggests that greater population structure and among population diversity at the trailing range edge than in the core, supporting this central marginal hypothesis. So now, using the same filtered data set, I asked, is there less genetic diversity within populations at the trailing edge as predicted by this hypothesis? So using the same data set with the highly admixed individuals filtered out, I compared heterozygosity, allelic richness, nucleotide diversity, and inbreeding coefficient, and I found no significant differences between these range positions in any of these comparisons, suggesting that genetic diversity has been maintained in these edge populations despite reductions in population size and loss of habitat. From a conservation standpoint, this suggests that for this species and potentially for other oaks that we are working with, range marginal position does not necessarily equate with deficiency in genetic diversity. There could be numerous explanations for this, and isolating the mechanism would require some more sampling, I think, and more work. But one possible explanation is that long-term introgression from overcup oak has infused genetic diversity into these Tennessee populations. So there are other explanations as well. So in summary, we found greater introgression and population differentiation at the trailing edge, but comparable genetic diversity when excluding hybrids, and actually greater genetic diversity when including hybrids at the trailing edge when compared to the core. So overall, this is providing some mixed support for this central marginal hypothesis in this case. So one of the key findings of this work is simply the documentation of a case of widespread cryptic hybridization between Quercus bicolor and Quercus lyrata. No reports of hybridization between these two taxa had been reported in Tennessee previously, yet we found it to be fairly rampant. And actually, there had only been a couple of reports of this hybrid at all in the past, and most of them were from the early 20th century in Illinois and Missouri. So this is a pretty interesting finding, I think. The frequency at which hybrids were found also suggests that hybrids are not strongly selected against. And we also found that hybridization events seem to be more likely to develop into this multi-generation introgression at the range margin. So overall, the evidence of advanced generation hybrids in these small populations at the range edge, combined with the finding that increased Quercus lyrata ancestry elevates heterozygosity and lowers inbreeding metrics, suggests that substantial levels of hybridization could possibly be mitigating the expected negative consequences of marginality by infusing these depauperate populations with genetic diversity and adaptive alleles. So in other words, where populations are small or isolated, these species can benefit by gaining variation from another taxon. An additional key takeaway from this is that these explicit tests of the central marginal hypothesis or conservation actions initiated at range margins should account for potential introgression. If not accounted for, results can be extremely misleading and could cause the inadvertent inclusion of hybrids into ex situ plantings. So some follow-up work that needs to be done includes better characterization of the morphological distinction between hybrids of various generational or backcross categories, essentially determining if hybrids can be reliably identified in the field. It would also be good to do more extensive sampling of Quercus lyrata dominant individuals to see if gene exchange is bidirectionally balanced or asymmetric. Also sampling seedlings from these populations might elucidate if hybridization is ongoing or if rates are changing over time. Furthermore, additional tests to determine if introgression is adaptive and evaluating the ecological significance of hybridization would be really interesting and are important areas of research much needed in Quercus more generally. There are also other parts of the range where hybridization might be occurring that could be investigated and compared with the results of this study. So these are just a few questions. There are numerous others worth exploring. And I will say this work is really just a first step and actually brings up a lot more questions than it answers, which has been really exciting for me to be a part of. Also, as a relatively recent initiate into the world of Quercus, being involved in this research has been a great opportunity to dive into all these peculiarities and the charisma of these oaks. So with that, I will conclude the talk. Here are some references. And thank you all for listening. I hope you found something interesting or thought-provoking. And if there are any questions, I think we should have some time.
Yeah, wonderful. Thank you so much, Jesse. Yes, we do have time for questions, so feel free to unmute or, yeah. Any questions?
I have a question.
Mhm.
Was wondering if this might have any implications for adaptation planting for climate change, such as, maybe, might suggest, choosing some seedlings or some, growing stock from the trailing edge of the population. Curious about that since that's work that I'm involved in.
Yeah, I think there are a lot of implications. I guess one being that if you were to proceed with this without like really knowing the genotype of the individuals, you might end up planting like hybrids all over the place without realizing it, which I think might be warranted and could be like potentially a good idea. But if you're doing it without realizing it, that might be a bad idea. But as far as like intentionally taking these kind of introgressed individuals and planting them elsewhere, I think that we don't have enough information on like the performance of these individuals like in a in like a controlled setting. So it's just it's a tricky question because I don't think there's enough information yet to know like if we should, because there are some pure swamp white oaks within these populations. So it's like, do we prioritize collecting seed from those or from hybrid individuals? I think without further testing, it's hard to say. But yeah, as far as does it affect that and have implications, yeah, but it's hard to have a clear answer at this point.
Sure. Thanks.
Great, thank you for the question. There was a question about Jesse, if you wouldn't mind if you could share your email for follow-up questions from folks.
Oh yeah, I can put it in the chat.
Great, thank you. And then there was another question in the chat. Have you tried applying structure analysis to a suite of morphological measurements?
So that's something that I want to do next. I haven't yet. So far, yeah, I haven't done any formal morphological analysis. Yeah, and I think that there's something there. For me, looking at these, it was just really confusing. I mean, there's so much variation that I was finding within each of these populations. So prior to having this, like the Q values for these individuals, it was yeah, it was just very confusing. But now that I have that information, I think we could make sense of the morphology a little better. So that's, yeah, it's a next step, I think.
Great. Thank you. All right. Thank you for the question. There was another one in the chat. So from Luciano, he said, I'm sure these interactions occur in several cases in Mexico, and it would be very relevant to study some of the most important ones. How valid is the morphological and distributional evidence to begin studying some cases? Or what are you basing your hypothesis on?
Sure, I fully understand the question. So why did I start studying this at all, I guess? Um,
I think, yeah. And I don't think his microphone's working, so Luciano can't.
yeah, so I guess, when I first started working with these um, populations in Tennessee, I did notice like some morphological oddities. Um, so they definitely appeared different, but I had no reason to suspect that like all of them were hybridizing. Um, so that was kind of what led me to, look into it. But yeah, I wasn't expecting like this level um, of introgression. And I think with different species, it's going to be harder to detect depending on how different they are morphologically. So I think, yeah, genotyping as many of these oaks as you can is the best course of action. Um, but yeah, going into this, I didn't really have a clear hypothesis about what level of introgression to detect. Um, yeah.
All right. Thank you. And yeah, Jesse's emails in the chat too. So if there's follow-up for that, Luciano, you can message him. Yeah, Roderick, do you have a question?
Yeah, I have a question or a comment. Your presentation brought to mind something that thought of and discussed before, that conservation efforts all seem to be aimed at conserving a species. We've got to conserve this species, otherwise we'll lose it. But it seems to me that what Quercus does, I mean, I don't want to personify it, but Quercus adapts to change by hybridizing, by getting alleles from other species, and those are better, more adapted. So should we not be conserving hybrids and trying to move hybrids to other places rather than focusing on conserving a species and accept that, well, we've got to lose this species because we're going to find a new one? Is that ridiculous, or do you think there's anything in there?
No, I don't think that's ridiculous. I think I think the narrative of hybrids being like this just sort of isolated, inconsequential thing is part of the problem. But when you're finding it across the landscape at like a really broad scale, um, that's what makes me think that like conserving these entities as they're genetically distinct, they have their own phenotypic traits, and that's probably influencing their environment in a lot of different ways and influencing the insects that are using them and all of the other creatures, and could potentially be shaping the entire ecosystem if it's like a foundational species. So ignoring them when it's, especially in this case where it's like a numeric majority of the populations that I found, I think is short-sighted. So I think conserving them, but it's just there's all of these barriers to that. There's policy and funding and that sort of thing, that it gets tricky. But I think we need to put more thought into that. Yeah.
Thank you.
Great. Thank you for the questions. Any other questions? All right. Any general questions about the International Oak Society or these webinars or the tours or events Roderick shared earlier?
I wanted to add a couple of things. that I have forgot to mention that there's a high possibility we'll look into a tour of Korea for the iOS in the future. So hoping that might work for 2027 or later. Also in iOS news, which is we've started announcing this at the conference and then in communications, but we have an updated Oak Names database. And Charles Snyers, who I think is on the call, has been responsible in large part for that. And he actually put something in the chat where you can look up the names of 10 named hybrids in South Korea. So if you haven't checked out the Oak Names database, please do. We've had it for a long time, but we've now got a new design, and it's much more efficient. So we're able to update it more quickly and efficiently. So I'm very pleased with that. So make sure you check it out because it's a good resource.
Great. Thanks, Roderick. All right. Any final comments or questions? Yes.
And thank you so much for your offer to suppose the visit to Korea to see oak in the wild. I think it's very meaningful for us because if you visit the wild and also to visit the herbarium specimen, herbaria, and you may understand much more on the oaks of Korea. So I would like to suggest to visit Korea, maybe next year or this year, to see oaks in the wild. Please come to visit Korea.
Great.
Yeah. Thank you, Yong-Shik. Yeah, that would be wonderful to have a tour of the Oaks of Korea next year. So we should start planning that, Roderick.
Yeah. I just put in the chat, there's a journal article, a trip report by Josef Suchek. This was in last year's journal. There's more information.
Yes.
Yeah, recommend that.
All right. Well, yes, thank you so much again to our presenters, Yong-Shik and Jesse. And thank you again for staying up so late, Yong-Shik, and everyone else who joined us, wherever you're joining us. So thank you again. Yeah, Roderick?
Yes, I would like to thank Amy Byrne for hosting this webinar. Thank you very much, Amy. And also to thank Morton Arboretum and the Global Conservation Consortium for Oak, who has actually provided the technical wherewithal to have this webinar. So thank you very much, Amy, and thank you to them and to our sponsors as well.
Yes, thank you. And
Thank you
thanks
so
everyone
much
for attending. Have a good evening, day, afternoon, everyone. Take care. Bye-bye.
Bye now.